Hi, I'm Dr. Arthur Brodsky, Assistant Director of Scientific Content at the Cancer Research Institute. And today I'm grateful to be speaking with Dr. Enrico Lugli, a CRI Lloyd J STAR at the Humanitas Research Foundation in Italy. Welcome, Dr. Lugli.
Thank you, Dr. Brodsky, for the invitation. It's a great pleasure to be here today.
Glad to have you. So as a CRI STAR, you're exploring one of cancer immunotherapy's central paradigms, that cancer-killing T cells can become exhausted and thus dysfunctional against tumors over time. But through checkpoint immunotherapy, we aim to overcome this exhaustion. And so along those lines, you recently discovered rare subpopulations of T cells that behave similar to stem cells. And unlike the T cells that can become exhausted, these T cell populations can maintain their ability to self-renew themselves and are crucial to the immune system's ability to eliminate tumors, especially over the long term. So what do we know about these stem-like T cells and the factors that determine the exhaustion versus self-renewal balance? And what more do we still need to know about them?
Yeah, so thanks for the question. This is a very critical point because it started more than 10 years ago when I was a postdoc, we identified a population of stem-like cells in healthy individuals. And then the work of many other people later on also found that in chronic infections and tumors, you find pretty much the same hierarchy of differentiation. So it was very good to know that we have a population which has exhaustion traits, but at the same time can self-renew and differentiate. And actually it turned out that this population is among the memory cells, one of the major targets of immunotherapy. So what we know is that this population can actually respond very well to anti-PD-1, for example. We know that this population can self-renew and act as a sort of progenitor of the terminally exhausted cells. We also know that these cells have at the same time, so this is critical because they have this sort of hybrid phenotype, because at the same time they have stem-like traits. It seems a paradox, but stem-like traits, but at the same time dysfunctional traits. And this is determined at the level of epigenetic modifications. So what we don't know, for example, is how these epigenetic modifications can be somehow overcome. Because one thing that we have shown, and also other people have shown, is somehow these cells, even though they have this stem-like fashion, they're committed to dysfunction. So when you stimulate them, when they respond to immunotherapy, somehow they're condemned to become terminally dysfunctional. And so it's difficult to have a function which is maintained in the long term. At least this is from the data in mice. The data in humans is much less clear, because obviously the number of assays that we can do is much more difficult. But for example, trying to identify, for example, what are the mechanisms that can overcome this imprinting in the population will be very important in the future.
And so, I guess the other side of the coin is, in contrast to these cancer-killing T cells that you want to minimize the exhaustion and promote their ability to self-renew, as well as be effectors that can go out and kill cancer cells. On the other side, you have regulatory T cells, or Tregs, that can actually help tumors survive by suppressing the immune responses against them. Do these Tregs also have different subpopulations that can either become exhausted or long-lived and self-renewing? And if so, how do they factor into the equation?
Yeah, so work from also our collaborators in Milan, but we have also advanced this even more, is we have a phenotype in tumors which is very different compared to the peripheral blood, for example, or other tissues. So for
Tregs?
About the Tregs, yes. So for somewhat reason, these Tregs are hyperactivated in tumors. They're constantly activated. And we have found heterogeneity. So they don't look alike. There is subsets, so there is different populations. We have found hyperactivated Tregs. We have found more quiescent Tregs. So that means Tregs that probably are bystander, but we don't really know very much. But a subpopulation looks very hyperactivated, and we have shown in collaboration, for example, with Axel Kallis at the University of Melbourne, if you remove some proteins, which are called transcription factors, these are proteins that control the genetic architecture of a cell. If you remove these transcription factors, actually this hyperactivated population disappears, and you have you can induce anti-tumor immunity, and that's very important. So that suggests that you have a target, or at least a sort of a number of genes that can be targeted in this way. So we have heterogeneity in that population, and Tregs, we hope, that will become the next target of cancer immunotherapy. So there is very much hope, at least from preclinical testing. There is now some phase 1 clinical trials. We don't have the data yet, but it looks like a very promising target to boost the immune response. And the reason is because this population is so much suppressive, and you find these cells pretty much in all the tumors, and they have more or less the same phenotype, even though you might have some differences from tumor to tumor. And if the behavior is the same, the important thing is ideally you could develop a drug that can target the same population in different types of cancer, as, for example, anti-PD-1 might work for different types of cancer. So it works in melanoma, it might work in renal cell cancer, it might work in non-small cell lung cancer. Obviously with different successful rates, but still the same drug ideally could be targeting the same population in different types of tumor, and that's the potency of immunotherapy.
That's exciting, and it makes sense that just as you'd want to support the good cells, you'd also want to block the bad cells. In this case, the Tregs that are helping to suppress the immune response against the tumor.
Right.
And so as you learn to potentially, as you learn what makes the cancer killing T cells behave in the way that you want them to, and what would make the Tregs behave in the way that you want them to, as you learn to kind of fine tune the molecular programs of these different T cells, how might this aid the development of future immunotherapy strategies, including cell therapies?
The field has advanced very much in the last decade. It really exploded. Was the capability to analyze one single cell at a time. So before, it was done at the level of bulk populations. That means you identify a population, you isolate that population, you characterize the population. Okay, so it's like taking people from a country, and then you compare it to people
trying to get like an average person that doesn't really exist.
Exactly. So in this way, we can see every single person. Okay, every single cell and the behavior of every single cell. So every single cell doesn't, so all the cells don't look alike, but they tend to cluster in subpopulations, and these subpopulations tend to have similar features. And if you can pinpoint exactly the specific population which is most important, for example, for responding to a tumor, or which is most important to suppressing the immune response, then you can have a more specific drug or a more specific approach to boost the anti-tumor immune response. So that's the important thing. And at the same time, you can develop more effective immunotherapies. So ideally, for example, in combination, as we have talked about stem-like memory cells, we know that those cells respond to anti-PD-1, but at the same time, for example, they have a lot of other proteins on the cell surface that we call co-stimulators. That means it's proteins that can help the immune response. And so ideally, if you use two drugs targeting, for example, PD-1 on one side and a co-stimulator on the other side, you could boost specifically that population, but not others that might not be very useful. So that's the beauty of the system.
Gotcha. And so as you pursue these goals, which, as from hearing you describe it, it's very it's easier to kind of look at the average population of T cells. It takes a lot more work to look at the individual one, but that's the kind of the precision that we will need to kind of get those to be able to help more patients. And so as you're pursuing this, can you explain why this CRI STAR funding is so important? And is this support enabling you to pursue things that you might not have been able to otherwise?
Well, so first of all, is very generous in terms of funding. So, and we are really honored to be supported by this important and international agency. So it was very honored and humbled to be judged by pioneers in the field of cancer immunotherapy. So that's very important. it's a great recognition also for the work that we have done in the past five to 6 years. This is just a premise that I wanted to give. The beauty of this funding is it gives you the freedom to pursue your ideas. It is not really tied to a specific research project. So obviously, when I submitted the grant, I had to propose an idea, and but it's not really tied to a specific research project. So, for example, if tomorrow we go to the lab, we do an experiment which gives you a result which is completely unexpected, and I have to say that most of the time it works like this. We can really follow that idea without thinking too much. Okay, so the CRI gives you that freedom. And for us, the freedom for our mind is probably the most important thing that we want to have in the lab, because we're really working at the edge of what is known and what is unknown. And when you enter the world of the unknown, you don't really know what you're going to find. And so sometimes the best discoveries were made by accident, in other cases because scientists made mistakes. And so we really want to have that freedom, and the CRI grants that freedom, and that's very important.
Yeah, I think that's a great point. Certainly that we have ideas, we have our educated guesses, and we look at the best available evidence and data to try to make a logical assumption about what the truth is actually going to be, but you don't really know until you start exploring. And like you said, that there's always lots of surprises. So that's great that you'll be able to follow the science wherever it points you.
Yeah, I just want to say that we're not really doing things by chance. Okay, so that's important to say. So we do something which is called hypothesis driven. So we have an idea, we work on the shoulder of giants, as other people have said in the past. We rely on the data that were published by other people, and we build on those data, and we make hypotheses. So we think that this is it's going to be like this, and then we go to the lab, we test that, and we follow the data. That's very important. So we follow the data and we follow the results, and we interpret the results by talking also with other people, with other scientists, and exchanging ideas. And then we think, okay, so it might be A or B or C, and then we try maybe all of them, and then we'll see what gives us the best and most interesting result, and then we go down that way. But I think it's important to have an open mind and not try to let's say to demonstrate what we think is going to happen. Okay, which is 95% of the cases it's not like that. So we need to be open. We need to be critical as much as possible.
And it's nice, as you mentioned too, that when an unexpected result does arise, or a new discovery or interesting fact does arise, you can pursue that without having to write new grants and wait to see if it'll be funded and all that. That's nice to be able to just go after it. So now, looking at the big picture, what do you hope to accomplish over the next 5 years as a CRI STAR, and how do you hope that your work will impact the field?
Well, first of all, I hope that it will impact the field. So. No, I mean, we want to invest our time and resources in a couple of ideas that you mentioned. So one side, try to understand what are the molecular mechanisms that are the basis of successful responses. Okay, so why patients respond to immunotherapy, which are the cells that respond to immunotherapy, and what are their characteristics. If you identify their characteristics with precision, then you can build new cells. What do I mean? For example, you can take that's something that we do all the time in the lab. So we take the blood of a patient, we can modify the characteristics of the cells, for example, by using gene transfer or by using small molecules, and sort of you can direct differentiation the way you like, okay, by using these tools. And then you analyze those characteristics by using technologies like genomics or flow cytometry and all these interesting tools that we have. And then we test these cells in an approach that we call adoptive immunotherapy. That's a very potent immunotherapy approach. So basically, you take these cells, you transfer them into an individual or at the preclinical level into you can do this in vitro or in vivo. And then you see how these cells can control the tumor. And you can mine these immune responses in this way, so by directing differentiation in one way or the other. On the other side, we have also the suppression effect that we know it exists, and it's very important. So we try to understand what are at the same time the characteristics of the suppressive cells. And in that case, we want to inhibit. Instead of boosting, we want to inhibit. So we aim to identify these molecular mechanisms. So on one side we can boost, on the other side we can inhibit, and for example by doing maybe a combinatorial approach, which should be able to have much more tumor regression than we're used to have right now.
Awesome. I really look forward to following your work and can't wait to see what you discover and what your work reveals. So thank you again, Dr. Lugli, for taking the time to speak with me today.
Thanks for inviting me. It was a pleasure.
Of course, and great luck with your work.
Thank you. Thanks again to the CRI.